Okay, so to start off, how'd you guys feel about the genetics exam?
I was not confident going into it, but as I was working through the problems, I realized I knew a lot more than I thought I did.
That's good.
I just gotta learn how to manage my time better.
Ah, don't we all?
Yeah.
I know, I've had those moments so many times. I'm like I focus on one thing so much to a point where I forget about the other thing I need to do, and then ah. But yeah,
That's how it's been with me for the online classes in general. Like I focus on one class so much, I forget like about cell bio or whatever.
mm, yeah, that can do that. It's just like you're putting twice the amount of effort for the online class itself, that it's like, oh, like you kind of get all over the place at that point. So this here is essentially how you guys kind of I guess ranged with the exam itself. This is actually based on all three of my sections together, which is pretty good compared to what I imagined it would be. So good job. If you guys are in like the down here area, let's try to get you guys more up. Okay. So, but don't worry, we have a lot more stuff that we can do to bring your grades up if you need to bring them up. More exams, more assignments, discussion. We definitely have some more stuff going on. So don't panic, like this wonderful little picture I found. Okay, so I realized recently that discussion is kind of going ahead of lecture. So I'm kind of changing up the discussion structure again. I'm gonna take out the discussion videos because at this point it just becomes more redundant, and I'd rather have you guys look at more problems than this at that point, because we do have our sessions here. It's going to be more of mix, like concepts, problems, concept problems, that kind of thing. So I'm going to take this out. It'll probably lighten the load for you guys too. So, and with quizzes, I won't be doing passwords anymore, just because I feel like that was a headache for everyone at that point. So, but everything else is pretty much the same. Okay. So the quizzes, quiz number 3, I noticed that some of you guys haven't completed it yet. So I'm gonna keep it open until tomorrow, 11:59 p.m. Make sure to do it if you haven't done it already. With quiz number 4, there's no password. This is gonna be like a check-in attendance kind of quiz. So it's more of me trying to get feedback from you guys. This is something I typically do after the first exam to see where you guys are at, what you guys like about discussion, what we can improve as we go further along. Okay? So that'll be due next week, Friday, not Friday, Wednesday. So that's what's going on with that. Do you guys have any questions before we go into presentations? Okay. Who wants to go first?
It doesn't have to go in order of questions.
Any order.
Okay, so I'll go. Um, So how do it says that the host
Oh.
disabled
Whoops.
screen.
I seriously need to remember to do that.
Okay. You can see it, right, I think.
Yep. Ooh,
Okay.
pretty.
Well, thank you. Okay. So mendelian genetics. So my name is Gabrielle Esposito. I had question number 3. So my question was a recessive allele in tigers causes the white tiger. If 2 normally pigmented tigers are mated and produce a white offspring, what percentage of the remaining offspring would be expected to have normal pigmentation? So the answer choices was A, 25%, B, 50%, C, about 66%, D, 75%, or E, about 90%. So some background information and how to solve the question would be, for this question, keep in mind that the capital T is the dominant allele and the lowercase t is the recessive allele. Hence, the recessive white color would be little t, and the normal pigmented color would be capital T, or capital T, little t. In the hybrid cross to the right, you can see the results when mating 2 normally pigmented tigers. So this is the monohybrid cross, and you can see here what you would get from crossing the 2 normally pigmented tigers. And then what this tells you is 25% or a quarter are white colored, so little t, and then 75%, 3/4 are normally pigmented tigers, big T, or big T. So that was also the answer to our question. And at this point, you have received your answer of 75%. And the end.
Good. Does that make sense to everyone? Are we all cool with this question? Okay. Who wants to go next?
I just have a question. I submit
Yes.
that presentation online, right?
Yes.
Okay, thank you.
Are the people who are supposed to go this week here? Because if not, I mean, I can go since you said I can make it up.
Okay.
You want me to go?
Yeah.
Okay. 1 second. Okay, so I was supposed to go last week, but I can't read. So this is what happens. Luckily, Nimi is nice and is letting me redo it. So my name is Paige. Last week I had question 3, which is, what is the outcome of synapsis, a significant event in meiosis? The answer choices are A, side-by-side alignment of non-homologous chromosomes, B, dyad formation, C, monad movement to opposite poles, D, side-by-side alignment of homologous chromosomes, or E, chiasma segregation. I chose D, side-by-side alignment of homologous chromosomes. I knew it wasn't A because the chromosomes are homologous, which means that they have the same genes, and non-homologous chromosomes have different gene sequences. And synapsis occurs in meiosis in prophase 1, and during synapsis, chromosome 1 from mom and chromosome 2 from dad, they come together and swap segments, and in order to do that, they need to align.
Good. Okay. I have Taylor and Aaron for this week. Taylor?
I'll go.
Okay.
It's not letting me share it. Can you see it?
Yes.
So I'm Taylor, and I had question 1, and it asked me that 2 black guinea pigs were mated and over several years produced 29 black and 9 white offspring. Explain these results giving the genotypes of the parents and the progeny. So I found that the mother and the father were both black and both dominant for black. And this is because, and this causes the offspring to have a ratio of 3 to 1, because the black color is dominant over the white one. And that was pretty much it.
Okay. Okay, so yeah. Yep. Does anyone have any questions? Concerns? I don't think Aaron is in today, so we'll just continue on. Let me just get back the PowerPoint. Okay. Okay, so we're gonna look into more of, so we start off with, so last week we had talked about Mendelian genetics and all the laws and postulates that come with it, right? So this time we're gonna focus on some concepts that don't actually follow Mendelian genetics and all that fun stuff we learned. So I'm gonna put on a video for you guys to just kind of give you like a little overview. If you guys are wearing headphones, make sure it's lower volume because this can get a little bit loud. Okay, I'm gonna start it.
I don't really think it's working that well.
Can you guys not hear it?
Yeah, I don't hear anything.
Okay, I guess we're not watching the video, but I'll put it up so you guys can watch it. It's really cool. I like it. It kind of summarizes what we will be covering. I hate Zoom sometimes. Boo. Okay. So we're gonna call this extensions of Mendelian genetics because it kind of correlates back on the concepts we covered with that. So with this, it belongs with okay, my words are not coming out of me today. Okay, so the first thing we involve with is gene interactions. This means that a single phenotype can be affected by more than one set of genes. So an example of this would be epistasis. I'll cover more of that in a second. So the next thing that involves with this is the alleles can be either incompletely dominant or co-dominant. In this case, with genes of different traits, don't always assort independently, especially when they're linked on the same chromosome, okay? So if you have like hair color and eye color on the same chromosome, then you won't see those two genes assorting independently, okay? So these scenarios kind of cause deviations from our expected Mendelian ratios of phenotypes that you see in the offspring itself, okay? So what is epistasis? Epistasis is when one gene pair affects or masks the expression of another gene pair. So for an example, so you have two agouti mouse. So the gene A involves the color, and then gene C controls if it has color or not, okay? So black is the recessive for A, okay? So in this case, you see nine cases where it's all dominant A phenotypes, where you have a dominant C gene going on. So when C is dominant, you have color. When C is recessive, you don't see color, okay? So in this case, you see dominant A and dominant C, you get agouti. With recessive A and dominant C, you see black, okay? And then when you put any of those traits with recessive C gene, that's when you see all of them being white, okay? So the phenotypic ratio in scenarios where epistasis occurs, it's a 9:3:4 ratio, okay? Does that make sense?
Yeah.
Honestly, no, I'm a little bit confused.
Where am I losing you?
I don't know, like I'm halfway understanding what you're saying, but then like some parts of it, not so much. And it's hard to distinguish between, like understand the full picture of what you're saying.
Okay, so the big picture in epistasis is basically just saying that one gene can control the expression of another gene. That's the main takeaway with this scenario. And it comes in the ratio with 9:3:4. Does that make sense?
Yeah, that's more simple.
Like that's the basics of it. This is just an example showing you that one gene can control how it actually expresses.
Is there another example?
I'll go into a question and then we'll, it'll probably become more clear, I hope. I wish that video worked, would made it life easier. Okay, hold on. Okay. Before I do that though, I have a couple of slides on the types of dominance. So once I get through that, we'll go into a question and it'll make more sense, I hope. So there are three types of dominance. The one that we've been talking about is complete dominance, where one allele just masks the other allele, okay? Then you have codominance. Codominance involves blending of two alleles. So it kind of becomes an intermediate version of that other allele, okay? Not allele, of that two alleles that are dominant, okay? And then with incomplete dominance, you have it where it occurs 2 genes and they're both expressed together. So to visualize this guy, so when you look into these characteristics, you want to go with the heterozygous guy to kind of fully understand. So with codominance, you see both of it. Okay, I think I have it here. This is the definition for this. That's the definition for that. So codominance, you see both alleles. Incomplete dominance, you see that intermediate mix, okay? Does that make sense? So
Yeah, but that kind of sounds like what they were saying before about the blending and how that was like not true about genetics. When Mendelian first came out with his, there's 2 pairs and they show one or the other, not a mix of both.
okay, so that is why this is called extensions of Mendelian genetics, because there are exceptions to these cases. That's kind of why we're kind of covering this after Mendelian genetics, because at the time Mendel was doing all of his work, he worked with pea plants, right? So when he worked with the pea plants, all of these alleles for the color or the height, they were all on separate chromosomes. And with this, in this case, we're talking about genes, not genes. For this case, we're talking about alleles. Like, so this is Mendelian version of dominance. But then further studies kind of showed that there are situations of codominance. So an example of codominance would be like blood groups, like the AB blood group, okay? And then because you see a mixture of this. So in this case, if you have blue and yellow, you'll see both blue and yellow expressed on it. Whereas incomplete dominance ends up with the intermediate version of it. Does that make sense?
Yeah, it makes sense. It just sounds like everything I just learned about Mendelian genetics was untrue. That's it.
Yeah, it does that. There's like,
No big deal.
it's pretty much how science works, you know? Like you find exceptions to the cases. Like one person might find this, but then the next person will come in and be like, oh, there's an exception to that postulate that you made. And just be like, oh, science, what can you do?
Is this true in all animals and maybe plants also? Like eukaryotic cells? Or
It should be.
okay. So like if you blend red and white, you'd get a pink flower.
Yes, there's an example for that in your lecture slides. It's called snapdragons. So these flowers, when they come in, it's like you have a red version and a white version, but then when you cross them, you'll see one white flower, one red flower, and 2 pink flowers. It's fascinating when you think about it. But are we cool with this part here?
Yes.
Yeah.
Okay. So what term is used to express the idea that several genes exert influence over the same characteristic? What do you guys think?
Is it co-adhesion or co-extension?
B.
It's B. Yes. So again, this is a definition. Gene interaction is essentially saying that several genes can actually come together and influence that same characteristic. Essentially, several genes interacting with each other to cause a certain characteristic to occur, right? Does that make sense?
Yes.
Okay, so this is a question with epistasis. The color of a sunflower pollen can be orange, yellow, or white cream. Crosses reveal that true breeding, orange is dominant to both yellow and white cream. However, a cross between true breeding white cream and yellow plants produced F1 plants with orange pollen. The F2 populations of this cross resulted in a ratio of 9 orange to 4 white cream to 3 yellow flowers. What type of inheritance do these results suggest?
Is it incomplete dominant?
So one of the main, oh, wait, hold it. Okay. It is not incomplete dominance.
Is it dominant epistasis?
Close.
It's called dominance.
It's actually recessive epistasis. You guys were close on there. Okay. So in this case, okay. So when you see a 9 to, so you're seeing a modification of the 9 to 3 to 3 to 1 ratio, right? So that tells you 2 genes are involved, right? So in this case, so we know that since these guys, these 3 here, okay, these guys here involve single genes. So that knocks 3 of these choices out, right? So in this case, since we have 2 genes, now we're left with the choices of dominant epistasis and recessive epistasis, right? So in this case, we know that the phenotype of orange only comes in when you have at least, so with this saying here is that when orange is dominant to both yellow and white cream, right? So in this case, we know that orange needs at least 1 dominant allele in each gene for it to occur. So in this case, the epistasis ends up recessive. Does that make sense? So that's how the answer becomes B. Okay. So a condition in which, a condition in which one gene pair masks the expression of a non-allelic gene pair is called?
Is that just dominance?
Okay, in this scenario, oh,
Oh, okay. Is it epistasis?
yes, which I know the question might be confusing a bit, but in this, when you're thinking about questions about extensions of Mendelian genetics, the word dominance alone doesn't exist. If it was referring to dominance, it would be like it would be referred to as completely dominant, right? Because if it said completely dominance, then it would also be an answer to this. And in one case, oh wait, hold on. I need to talk about this. So we're talking about gene pairs. Dominance talks about just two alleles, right? When we talk about dominance, we're talking about the different alleles. Epistasis is two different genes controlling each other or masking each other. Does that make sense?
Yes.
Okay. If this thing likes to work. So the answer to this is epistasis. So it's the two gene pairs controlling one another, okay? Okay, so what about this guy? What is the difference between incomplete dominance and codominance?
Is it A?
A.
Yes, it is A. So this is just definition again. In codominance, it's both expressed, like that little cartoon I showed. In incomplete dominance, it's intermediate, okay? With incomplete dominance, a likely ratio resulting from a monohybrid cross would be what? If anyone tells me C, I will run away. Not C.
Is it E?
Yes. Okay, so with incomplete dominance, you have to remember so when you're thinking monohybrid cross, you're thinking, so the genotypic ratio is 1 to 2 to 1, right? In a regular monohybrid cross, right? And you get a 3 to 1 phenotypic ratio. So in this case, with incomplete dominance, this is the phenotypic ratio and genotypic ratio because you get one homozygous dominant, two heterozygous, and one homozygous recessive. And all three genotypic classes show different phenotypes, right? Does that make sense? Yes, no, maybe so.
Yeah.
Okay. So in ABO blood types, all of the following offspring are commonly possible except what? So what's going on here?
Is it B?
Yes. Yes. Okay. So that's B. You would never get an individual with type O from a mother with type A or AB because O is recessive compared to A and B. AB is codominant, so you'll see both antigens of those guys. Are we okay with this question? Guys,
Yeah.
okay.
Yeah.
So, so the next thing we're going to cover is more sex determination of an individual. So with animals or plants, the sex of those guys are determined by the X and Y chromosomes, right? So with Drosophila and humans, we see the XY, XX chromosomes. So females have the XX, males have the XY, and we're all happy. So when we're looking at this, we see that females are referred to as homozygous, whereas males are more hemizygous, not heterozygous, okay? So next, one of the things we look into is like X linkage. X linkage just involves genes that are present on X chromosome, and it exhibits patterns that are different than the autosomal genes. In the Y chromosome, it's relatively inert. It's male-specific genes that are involved in the human Y chromosome, and it lacks genes that are found on the X chromosome itself, okay? So a question here would be the inheritance of a skin condition in humans. Consider the following family history. Bob has a genetic condition that affects his skin. Bob's wife, Eleanor, has normal skin, and no one in Eleanor's family has ever had the skin condition. Bob and Eleanor have a large family. Of their 11 children, all of their sons have normal skin, but all 5 of their daughters have the same skin condition as Bob. Based on Bob's and Eleanor's family history, what inheritance pattern does the skin condition most likely follow? Okay, I know this question is a lot of words, but what do we know so far from this question?
Bob has a skin disease. All of his daughters have skin disease. None of his sons do.
Okay.
So it
So-
can't be B then, right? Because it's not Y linked if the daughters have it.
Is it
Is
A?
it the X? Is it C?
It
You
has to
guys
be.
are good. Okay. Okay, wait, not, hold on. Let me just double check something. Okay, you guys are right on the part where it's X-linked. Okay,
So maybe it's A because one of the parents have it, and we learned that if it was recessive, both the parents would not have it. So maybe it's dominant then.
you guys are amazing. I love it. Okay, so yes, so we know from the question, we have an affected father, a normal mom with no family history of it. And we see that all the daughters are affected, whereas none of the sons were. So this, as you guys said, it should be screaming X-linked, right? Then you see that it's shown in directly the next generation. So you know that it's dominant, that one allele of this trait can actually cause the condition. Now, what would we see if it was recessive? We wouldn't see the daughters affected, right? because they would end up carriers. And then the next generation males will end up with the disease because the X chromosome can, it's a half and half kind of situation there. Does that make sense? So that's why the answer is A. Okay. Duchenne muscular dystrophy is caused by a recessive X-linked allele. A man with this disorder would have what? How would this man inherit it or give it? What do you guys think? So what do we know?
Is it B?
Yes. So recessive X-linked. We know that since it's X-linked, we know that the man must be carrying the X chromosome that he got from his mother, which is the only place a male would get X chromosome. So essentially we have it inherited Y chromosome here from his father, and he would pass that X chromosome only to his daughters. So in this case, you would see that the guy with this disease inherited from his mother because it's X-linked, and that's where you would get the X chromosome. Are we cool with this question? Okay. So another thing to talk about is pleiotropy. Pleiotropy is essentially when one gene gives rise to multiple traits. So completely so it's essentially these three traits could be completely unrelated, and it's coming out of this one gene. Okay. So an example of this guy is Porphyria variegata. So in this case, you have, so Porphyria variegata is a autosomal dominant disorder that results in the buildup of porphyrins. And porphyrins are actually a precursor for heme, which is part of your blood, right? So with this gene, when there's an issue with this, it kind of gives rise to different phenotypic effects, like abdominal pain, then you go into muscle weakness, fever, hallucinations, so on and so forth, completely unrelated effects, but it's all caused by that one same gene. Does that make sense? Okay. So the next thing we want to talk about is sex-limited inheritance versus sex-influenced inheritance, okay? So in sex-limited, it's, as the name says, it's limited to one of the sexes. Whereas sex-influenced, you have the sex of the individual, itself influences that expression of that phenotype, whatever you're looking to. And sex-influenced inheritance is not limited to one sex or the other, okay? So an example for this would be in chickens and their feathers that they grow. So in this case, we have the hen feather is going to be controlled by a dominant H allele. The rooster here gets the recessive H allele, okay? So in this case, so we have the 3 genotypes. This expression of the sex-limited inheritance ends up being controlled by the individual sex hormones. So in this case, you see if you have the genotype of these guys all here, if you have a female chicken, that chicken will have hen feathered. Whereas if you have one in the rooster too, in the male chicken, you'll end up with hen feathered. However, if you have the recessive alleles and you have the high levels of the male hormones, you end up with this type of feathering for that rooster. Does that make sense? So this is specifically like depending on which sex that individual is, okay? Sex influenced. An example to this is male baldness, okay? So in this case, we have the B allele, the big B allele that leads to baldness. Heterozygous results in baldness in males, but not in females. So you can see that there's an effect depending on the sex of that individual, right? So, in this case, it would be like, so dominant, you can see it in both females and males, but it's more pronounced in male, okay? Whereas with this, you don't see it with heterozygous in females. Heterozygous in males, it's automatically bald. And then the recessive alleles, you don't see baldness at all. Does that make sense?
Yeah.
So, oh, somebody say something? No? Okay. So that is pretty much what I have for today. Do you guys have any questions, concerns? No? Okay, so that's all I got. The quiz is gonna count as your attendance and your quiz, obviously. So make sure you guys do it. And yeah.
Thank you. Have a good night.
You too.
Good night.
Good night.
Night.
Hi, I just have one question.
Oh, hey,
Um,
Quentin.
Hi. What was the password for quiz 3?
So that's actually within the discussion video itself.
Uh-huh.
If you see it in there.
Okay. All right, that was
Like,
it. Thank you.
Yep, okay.
All right, have a good night and
You
afternoon.
too. Everything all right, Siobhan?
Yeah, everything is okay.
Okay.
Thank you.
Have a good night.
Yeah, same to you.